evolution has built all the anti-aging mechanisms that we have. So it's not really a question of why do we age. The question is, why aren't we even longer lived? Or why are we even as long lived as we are? And the answer is, well, it's because there's a bunch of active mechanisms that give us this longevity. So fundamentally, if you want to push this substantially further, you need to put in new mechanisms and new information. And by that, I mean like actual new systems in your cell that do something that never has been done before inside a human body.
Hi, everyone. This is episode 15 of the Polling Principle. Welcome. And today I'm very excited to have Michael Florea with me. We've known each other, I guess, for 2 years, 3 years from the longevity world. I'd love to begin with, maybe a little bit, what's your background, how you got into the, longevity world, what was maybe your training and what are you working on at the moment?
Yeah, for sure. And Javier, thank you so much for having me on. It's very interesting to speak to you every time. Uh, well, there's a short answer and then the long answer. So I suppose the short answer is that when I was young, when I was 14 or 15, I tried to find a purpose for my life. And I found, of course, the longevity space. And I also found the genetic engineering space. And I thought that bringing these things together would allow us to make, a very large impact on human health and life. So when I got into high school, that's when I started doing genetic engineering and biology, specifically towards engineering longevity. And then that took me through through different academic settings to my PhD, where I was working on gene therapies for aging. And in particular, I was building a whole body gene delivery system and then using that to build these therapeutics. And then finally, that experience took me to my current startup, which is around automating animal research. Because as I was doing these studies in my PhD, very, very clearly that became the biggest bottleneck, which was just a measurement of and throughput of these studies.
Cool. Lots of things to unpack there. So let me see if I can go through all of them. So um, uh, genetic engineering, of course, is maybe one of the most powerful ways, I guess, to change, how a human body works, right? Like the why genetic engineering versus the small molecules versus other therapies, right? Like, what is, what are maybe some of the pros and cons of that approach versus something else for longevity?
Well, I think they all have their place, and small molecules is where things definitely are going to start. And we have the first wave of longevity therapeutics now out there, arguably. Um, the issue with small molecules is if you play the tape forward in time, like into deep time in that sense, then very quickly you realize that they are going to hit a limit that you practically cannot overcome. And there's two limits there. The first limit is, let's say if you take, two or three drugs, usually, well, you're probably fine. They're probably not going to interact a lot. You don't have to worry about that. If you take 10 drugs, now you definitely need to start worrying about interactions, even, chemical interactions. Just scale that to 50 or 100, you know, 100 drugs, factorial of that is, about like 10,000 interactions that you need to worry about. And so it very quickly becomes unmanageable to deal with the potential side effects that comes from so many things. So that's one issue. The other issue is just, I suppose, the practical issue of, longevity is a function of doing something every day in terms of both prevention and treatment, and adding an extra drug to your regimen every day with that compliance means that at some point you just can't do anything else except for just thinking about what drugs you're taking. And I suppose that's what we see in Brian Johnson, who's like really taken to the max. And of course, that's awesome what he does, but it's not a lifestyle that most people can or want to sustain. Um, so that's my perspective on small molecules, and in fact, pretty much any therapeutic that doesn't provide a lasting impact,
Mhm.
because you just hit these scaling issues. And now the question is, well, what would provide these lasting impacts? And there's really only a very few things. And, arguably replacement is one of them. Like if you switch out an organ, well, that organ is going to be better until it gets worse. So that's pretty lasting. That's one. But then genetic engineering or modifying the actual behavior of the cell is another way, and arguably the way to do that, because of course we humans as a species, we are not the longest lived species by far. There's ones that live much longer, and these animals don't do any medicine at all. They don't go to the doctor. They have these systems encoded in them that allow them to live longer, and that's, I think, what we need. It's that that's the way to do it.
Yeah, yeah. I mean, I like to think about it as, I mean, I don't know if we go up to the automobile analogy, right? Like you can take a car and then you can uh change the filter, I guess. You can clean it up, you can I don't know, you can kind of adjust the pieces so that they fit properly or whatever. But um maybe longevity is a problem that can't be tied down to just maintenance, right? Like maybe there are other things that you need to do, right? Like, so at some point, if you have a car and you want the car to run faster, you just need to design a new car, right? Like you
Mm.
need to replace, one engine with another engine. You need to replace, one, suspension with another one or whatnot. Um, and I guess, in my mind, genetic engineering, it's a little bit like that. Like you're basically changing the behavior of ideally every single cell in your body, right? But it's not the small tweaks that you can do with small molecules, right? That just stimulate something or inhibit something, right? But it's like you know drastic potential drastic changes, right?
Yeah, and the way that I think about it is, well, the question is, why do we age at all? And the answer is very simple: is because evolution has built all the anti-aging mechanisms that we have. So it's not really a question of why do we age. The question is, why aren't we even longer lived? Or why are we even as long lived as we are? And the answer is, well, it's because there's a bunch of active mechanisms that give us this longevity. So fundamentally, if you want to push this substantially further, you need to put in new mechanisms and new information. And by that, I mean like actual new systems in your cell that do something that never has been done before inside a human body. And it's just fundamentally that's what you need for many of the things that we know go wrong in aging. And, really genetic engineering is the only thing that allows you to do this.
And I guess the thing that is exciting about replacement as well is like, in principle, an organ is a nice abstraction, right, in the computer science sense, right? So if you could somehow take one out and then put another one, and then, all the interfaces, all the vascularity or the nerve or whatever, right, the nerve endings, if you can replace all the stuff properly, right, then you get something that works. But and I guess eventually you could also improve function, right? Like so, if you're replacing a kidney, for example, and you put another kidney that is better at filtering out, some toxins, for example, that human kidneys are not good at, right? Like you could get not just replacement, but also something that is, superhuman kind of abilities.
Exactly. Exactly. And you can do the same thing with genetic engineering also. And I think both of them are definitely very valid approaches. And I definitely don't want to, I mean, I, the replacement should definitely be explored. But when you think about from almost like a practical perspective of what do you personally want to do? Like, do you want to go and have a surgery every couple of years? Most people don't, right?
Yeah.
And then, like, how do you manage the supply of growing all these organs and just making sure that they're fine? And it's just there's another series of logistics issues there. And of course, the ultimate level of replacement is whole body replacement. Like, it's just transfer your brain from one to another. And you can argue that maybe you can replace the brain as well, though. It's a different discussion. But it's it hits the same issues, which is like, how do you make them? Uh, are you can you even overcome the potential like social resistance to this, which could be much more difficult to overcome than the technical issues. Mm. And even then, assuming that you can't replace the whole brain, you are fundamentally limited by how by much the rest of the body could then rejuvenate the brain. which would probably still be quite a bit, but not infinite.
Yeah.
Well,
I mean, I guess even the brain, you could, I guess, replace it slowly, right? Like one graft at a time. I think that was like what Gene Havert and I guess.
well,
It-
yes, and this could work. And again, I think it definitely should be explored. But when you do this in a mouse, the size difference between a mouse and a human brain is 2,000x. And we're talking about being able to grow maybe like a millimeter thickness grafts at most, and you can't take out too big of a chunk of the brain at the same time either.
Yeah.
So, like doing 1,000 surgeries on your brain isn't going to be practically feasible or something that people want to do. And I think that's where the importance of the scaling issue comes in, which is, you know, yeah, just like volumetrically, it might not be possible.
Yeah. Yeah. Kind of reminds me, there was a movie about this whole body replacement. It was called The Island. I don't know if
yeah,
you watch it.
yeah, I've seen it. Yes, yeah, yeah,
So I guess for everybody else, it's I guess Ewan McGregor and Scarlett Johansson, right?
yeah,
It was. I mean, it didn't really go into any of the actual morality of it or whatnot, right? But it was a cool action movie.
mm,
Um, Cool. So then, what is the, I guess, translation path, or what is the rationale between saying, okay, let's do all this, amazing, genetic, engineering in, humans' health, to like, well, let's, create a company that improves
uh,
animal experimentation, right? Like, what is the path?
yeah. So there's a couple of paths that all converge together. But the first part of the story is that when I started doing this as a teenager, one of the things that I very clearly realized was it's gonna take a really long time. Like to be able to do genetic engineering for human longevity in humans is a 30-year time horizon where you have to build everything, and it's gonna be technically very difficult. But if you're there, you can have a very large impact, but it takes a lot of engineering to get there. So that's one. And particularly what we're missing is we're missing ability to create better tools more rapidly. Right. So right now we don't have good tools, but we don't also have good ways to create better tools fast. And arguably the best way to accelerate this is, to create a tool that allows you to create a better tool faster and, just keep on going until you get to the place. And that second aspect is what came out of my PhD very clearly. So in my PhD, I built this system that is able to hit at least 80% of the body mass in an adult old mouse consistently and in a way that I can control to a certain extent how much each tissue and organ get that DNA. And so right now it hasn't been published yet. I mean, we're just about to get this out. But the summary is that um, I did something in my PhD that many people thought was just not practically possible, or at least not feasible, which is to hit the whole body and convert it into genetically modified it. So I got the system done, but then when I tried to test the effect of whatever I was putting in, that's when I hit a wall. And the wall was, for example, if you want to know where your DNA goes or where your protein is being expressed, you have to take out that tissue, you have to process it, then you have to analyze it and run it, run with that. If you do this at the body scale, even if you don't take all the organs, but if you take like the major organs, you have to take about 25 organs and do, DNA, RNA, and protein pretty much on all of them. And right now, the throughput of doing this, just the manual labor, so slow that you just can't get through it. It just one iteration cycle is just too long. It's like a month or maybe 2 months, right?
Or
Yep.
just to do like I guess I don't know how to call it, like the autopsy, of a mouse, and then cutting all the different tissues and then running them through the mass spec, or like how do you do that?
So yes, so basically the way that it would look like is if you wanna for any drug, this is true, but particularly for gene therapies, if you wanna hit the whole body, which for longevity you have to do. So you wanna know where your DNA goes or your drug, you have to inject this into an animal, usually it's a mouse. Then you have to euthanize the animal and harvest the tissues, and then analyze each tissue for DNA, RNA, and protein expression, right? And for it's going to be your viral DNA or your genetically modified DNA, and then your protein and RNA for whatever gene you're trying to overexpress.
Yeah.
Now, for one mouse, it's easy to do. It's like maybe 25 tissues, you run them, and then you do these 3 separate tests. But one mouse is never enough for an experiment. You usually have to run at least, let's say, minimum of 6, maybe 10, more like 20 or 30 if you really want to do a proper study. And right now, the toolkit that we have just to do these basics, 1, 2, 3, is still like very manual. You have to do everything almost by hand. Like many of the steps are done by hand. And just the throughput of testing, like what actually works better and so on, just isn't there. Like
Yeah.
no one had built that pipeline. So that's one.
Right. But the sequencing part, I guess, at least for the RNA-seq, that part is vast now, right?
Yeah, like that part is not the issue. Like once you get the stuff into the machine, they're pretty good. Even with the data, even that is, I suppose, more difficult to do than just doing the run. But let's say you have an RNA-seq data. If you have a pipeline, you can also analyze that pretty well. But you don't actually want to run RNA-seq for most things. You want to do something like qPCR or something more targeted, more low cost. And then it's also like more manual to set things up. So it's like the whole like workflow closer that one. But even more so, I spent my multiple years doing this, figuring out the correct systems and everything. Got to a point where I had a system that was working well enough so I could take either naturally aged mice and try new gene therapies on them, or take a disease proteroid mouse and try to like fix it, actually generate a treatment for it. Right?
Mhm.
And then what I discovered was, well, if you do something like a longevity therapeutic, theoretically, you could only measure one thing, which is lifespan. But that's not good enough because it takes a whole lot of time. And if things don't work, it takes you forever to find this out. And even if things work and you have an increased lifespan, you don't know if you actually made the organism more healthy. There are drugs that actually increase lifespan, but make the mice lethargic, or
Yeah.
or give them
Mess it
better
up,
movement,
right?
whatever. Yes.
Castration, I guess, was one of those things. I was like, yeah, doesn't translate to humans, right?
Well, it actually does. I mean, castrati are log 11.
But I mean, yeah, it doesn't translate. I mean, like nobody would go through it, right? But like, yeah, it translates in the sense of like, yeah, like as a treatment, it could work. Yeah,
No, maybe we go and pitch that to Brian Johnson.
may in the scene. And he may, give it a thought.
Yeah.
Oh, okay.
So you have to measure health in addition to pure longevity.
Mhm.
But the way you do that now, and how I studied in my PhD, was for every single metric, you take a mouse, you manually put it on some test setup. For example, for strength, you like hold it by the tail, you let it grab a beam, and then you have to pull the mouse just the right amount so that it kind of holds onto the beam. Usually it doesn't like holding onto the beam, so you have to do it like 10 times over. And then because it's noisy day, you do this another like 3 or 4 days, and then you get your measurement. And do it for every single mouse, and then you do it for every single test kind of thing that you want to do. For example, if you want to measure their endurance, you have to do this on a treadmill and just like watch them run for 2 hours and a treadmill, and they take it off. So every single measurement that you want to do on actual health, that is not just like like a blood test, is super super laborious, and it's just like super basic.
Mhm.
And I spent about a year in my cohorts where I treated 3 or 4 different therapies that I tested, just doing these manual measurements all day, every day, just to get the very basic data. And even then, it wasn't good. And guess what? I couldn't replace myself, because when you as a person do the metrics, your hand is going to impact on the measure. So if you now want to compare 2 data points and you switch the person in between, you're going to get different data just because it's a different person.
Yeah.
No, once you start this, you're locked in basically for doing these manual measurements for like 12 to 18 months.
Yeah.
And it's just insane.
It's crazy. Yeah. I do remember last time that I was talking with Noah, I think it was,
Yeah.
with your co-founder. He was telling me about sometimes people would run experiments and maybe one section of a lab have a window that was open and then the other section didn't. And then a result would not replicate because one of the mice was next to the open window, got fresh air every day, right? That the other one didn't. And then you think like even the most minute, differences, right? Like, of course, like if you know how to handle the mouse properly and then an undergrad comes in and then replace it, doesn't know how to do it, that data is just not going to work.
Mm, mm. Well, yes, but even then, it's like when you look at different labs, you have highly experienced people that are just doing things differently. And we tried outsourcing one of our studies to like a CRO or contract research organization. They
Mhm.
switched technicians midway through the study. And when you clustered, biggest effect was of the technician. Right. And so yeah, so that's very clearly in my PhD, as I thought, first, there wasn't a tech stack for pure delivery, pure genetic engineering in humans, that didn't exist. Secondly, the tech stack for developing that didn't exist. And the third, what I discovered was, well, you can't even test. Like once you have these first two, like you can't even test. And maybe the third one is the biggest issue because like if that's not there, you just don't have the throughput to do the other two. Um, so that's how the company started. Basically, I realized this was a fundamental issue that really held back the longevity field. And now to put this into context, if you don't have your own time and labor available and your own lab space and facilities and everything, if you want to outsource one of these studies, which is like a basic longevity study with health metrics, starting
Mhm.
from 18 months old, for like 100 animals, which is how many you really need to see as some statistical significance, guys. Yeah. Uh, it costs you 500,000 right now. So
You
what?
really need 100 animals? Like, why so many? They're genetically very similar, right? So you can't have, I guess, that much difference, or I don't know.
Yes. In fact, to do. Even if you don't do anything, there's substantial variation, particularly when it comes to aging. So that's one. By the time that they get too old enough that you want to measure them, there's just a lot of variation. And secondly, and I can go into why there's the variation, the where it comes from, but it's there. And secondly, more often than not, the effect size of longevity, like an individual longevity intervention, is pretty small. Like good one will have maybe 20% effect size on longevity or like some of the metrics. Most of them are like maybe 10%. But if your career is dependent on having some success in that 10%, that you say, look, if there's a 10% difference, then I can detect this, and you work for 3 years for this, then you better have enough animals to be able to pick that difference apart. So it comes down to a large variation and a small effect size of most longevity drugs.
Yeah, yeah, of course. But like the basic statistics, right? Like the larger the sample size, the more the power of that the statistical test, right?
Exactly.
Hmm.
Exactly. Yeah.
Interesting. Okay, so then I guess from the perspective of uh, a tool, right? Like what you're building is something that would enable people to do that, but a much lower cost, right? Like is that the major dimension that you're trying to reduce, try to do same thing but much cheaper?
So the cost is one thing, but like all the three barriers have to come down, and the three really large barriers are throughput right now. Even if you wanted to do many studies, you couldn't because you just don't have enough hands available to. That's one. Secondly, the cost. So I mean, you could do many studies right now if you outsource them, but most people just can't afford 500,000 for like one basic study. And then the third is the rigor or reproducibility. And there, whatever you're getting today from that one person, you should have confidence that you can interpret this and repeat the same thing if a next person comes in or from a different lab, but just doesn't fundamentally exist right now in the animal research world.
Yeah. Um, and maybe this is kind of already a little bit obvious by now, but like why mice versus other animals that may be easier to kind of, put into a little plate and fully automate, with a robot or whatever, right? Like, I mean, like a few weeks ago, I spoke with Ben Blue
Mhm.
from Auto Biomedical. Of course, they're using mice. I had a conversation a while back with somebody that was using zebrafish, which have a nice benefit that they're sometimes transparent, so you can
Mm,
see inside of them, right, or
mm,
whatnot. You could use, monkeys, I guess, but monkeys would be much more expensive and harder to get, and they have other regulations and whatnot, right? So like, do you use mice because is somehow, easier or cheaper, or because that's just what people use in longevity, right? Uh,
That's just what people use in longevity and across the entire biomedical research space, really. It's about 90% of all research animals are mice, and another maybe 5% are rats, and then everything else makes up the rest.
uh, um, but then, yeah, I mean, sometimes things translate from mice to humans, and sometimes they don't, right? I guess it's a model like everything else.
Yes, and I think that's the other big area, which is, first of all, mice are like, you have to know exactly what you are using your model for and why. So, for example, if you want to study basic biology of aging, just want to understand the mechanisms, mice are a great model for this because they're most human-like, and you know, by and large, they still age the same way as humans do, although they don't necessarily die of all the same things as the major causes of death. But you can study like the fundamental mechanisms, and there's so much work to be done there still. In fact, I would argue that you can argue that pretty much everything, or most of the things that we know about aging right now, have come from most longevity studies where someone has made some effect and said, look, this mammal here now as a result lives longer. That's when people believe it, right? Yeah. So that's one. But I think the other thing where people go off is when they try to use mice, particularly poorly generated mouse models for specific human diseases, for testing a specific therapeutic, and they haven't done their legwork and their proper preparation to make sure that this actually replicates disease, the mechanism is the same and whatnot, then of course it doesn't translate. And that's because, well, guess what? Yeah, you didn't choose the right model. And I think that comes down to one of few things. There's first of all, there's I suppose, like, perverse social incentives around making stuff work in mice and then getting it pushed up, because that's when you can get your next paper or your grant funding or your raise or whatever, right? And so there's an incentive to take the easiest mouse model that will definitely work and give you some data, and then just like, oh, this is working, let me get
Mm.
my reward from this. I think particularly clear this is in cancer and
Yeah.
in neuroscience, right? So that's one. So people, they want to do this. They actually want to fool themselves in some ways.
Yeah.
Um,
Well, but in cancer, right, like you could even take a tumor from a human and then graft it in a mouse, right? And like, I mean, I don't know, I'm not an expert, right? But like, yeah, it sounds to me like that would be a reasonable model, right? It's the same kind of tissue, right? So like, if you have like say three cohorts, right, and like, or like maybe two cohorts, and one of them has that same graft, and the graft survives for 2 months, and then in another one you apply your treatment, and then the graft dies, right? Like, wouldn't that be a good model for, your drive actually killing that tumor?
you might think that at first, uh, like at first blush, and that's what many people do. In fact, I would say most people do that. And I suppose maybe there's some value there in like highly like precision medicine, where you take really the exact tumor from a person and then you try to figure out what drugs work on them. But the fact is that um, it's kind of a vanity test in the sense that when you try to treat a tumor, first you're putting a tumor into a foreign organism right off the get go. It's foreign antigens, foreign everything. If the mouse had a full immune system, it can probably deal with the tumor right there. That's one.
Yeah.
Oftentimes,
You're talking in the control too, right?
sure, right. But the fact is that the fundamental difficulty with tumors is that they are your own genetic material. But now you're putting it into a totally different organism, and you're asking, well, for it to, is it gonna kill it? Yeah, of course it's gonna kill it, right?
Yeah.
So I think that's one. Then secondly, the immune systems are different, very different between humans and mice in I know sometimes. So at the very least, if you do that, you would have to also humanize the mouse immune system by removing the blood cells, putting in human blood cells, and that's very doable. I mean, it's not easy, but it's doable.
Yeah.
And our
Yeah.
lab,
Passive, I guess. Right.
yes, to my surprise, very uncommon done in the drug development space. So using mice with humanized immune systems is not a common thing, period. And then there's a whole bunch of other things like genetically now, you could be modifying the mouse or it. be your T cell system to be more human-like, or like whatever. You could just make it much more, or just use the mouse tumor and generate a more human-like mouse tumor that's now in the thing. People just don't do it. And that's because they know, well, if I generate this model, it's going to take me longer. There's a lot lower chance of working because actually it's more difficult to solve. Therefore, maybe if I run out of time, then I don't get my result and I have nothing, right? So I think they want to default to a certain extent to this thing, to this like simplest model, right?
Yeah.
So that's one. But I think the other big part there is, look, I think we can make mice work much better. And, forget about the fact that even the basic studies right now that are being done in mice, we don't know if they replicate because one human does it one way or another does it in a different way. And it's written down on a piece of paper and it's just like, it's totally not even there. So let's say we solve this, we improve some of the basics of reliability of the mouse testing or animal testing period. Well, the next step of that is humanization, of course, and actually generating better models. And here, the same toolkits for human whole body genetic engineering would work particularly well also in animals, because if you can quickly generate the genetic model rather than having to wait a year and a half when you know do the germline edit and then breed them and then back cross them and then you get your homozygote animal that has both mutation, then many more people would probably do it. They just say, look, I generate my models and here they go, right? So that's the second step. And now the third step is, but look, mice as a species probably aren't the best model for many diseases, particularly complex diseases. Some things you can measure in vitro, but actually not most. Maybe rat, but
Degenerative diseases, right? Like anything, psychological diseases, right? Like,
exactly.
none of those
Yeah.
work, right?
Yeah. But people don't use other models because the genetic modification tools just aren't there. That's one. And also because, secondly, because they're more expensive.
Yeah.
But I would argue that actually it's the fact that you can't generate, in many cases, a specific genetic disease, or you can't just replicate your model very well in something that's like a monkey or a rat even, because if you want to make these modifications, it takes you 2 or 3 years to just breed them and get your animals. So before we go into humans, you could take these other species and deliver DNA, use that as a model, and now bump up your predictability even further and, kind of take it step by step.
Yeah, yeah, interesting. A couple of comments that come to mind. One of them, I really like the idea of improving the reliability. I imagine, I've heard from other people like this is a big issue in scientific discovery, right? I think somebody was saying not long ago that about half of papers in the life sciences do not replicate, which is completely crazy. And like last year, we had this massive scandal about the big Alzheimer's paper, right? That was basically falsified data, right? Uh, it's one of those things that, um, a lot of people don't know, for example, for like in the industrial revolution, right? Like, in manufacturing, of course, a lot of people will think of, well, like what changed was energy sources, right? You had steam instead of, like human or animal labor, right? But the other thing that made a big difference with manufacturing is reducing variability, right? So like if you study a little bit of like, good manufacturing techniques, right? Like, Six Sigma or whatever, right? Like it's all about making sure that the output of the process is the exact same thing over and over and over again, right? So like the whole Six Sigma came from like, six standard deviations outside of the mean, right? Like if you're making, trinkets and the trinkets was to measure, 1 centimeter or whatever, right? Like it has to measure 1 centimeter, right? It cannot measure, 1.5 or 0.5 or whatever, right? So like you want to reduce the variability of that thing that you're measuring, right? As much as possible. And that's what enabled, building, complex manufacturing, right?
Mm.
Um, the second thing that comes to mind is I was listening to a presentation by, I think it was Regeneron not long ago, and they were talking about how they had built all these, fancy mouse models, right? I wonder what you think about that, like, is that valuable or not, right? Because like they seem to be like one of the few people that have built this really complex models, but then they haven't come out with like a whole lot of new drugs, right? By having this technology. So like maybe is it too early for them, or like, they haven't, leveraged that enough, or like, where would that be?
I actually saw a presentation by, I think the CSO of Regeneron, George Yanopoulos, I think. I'm probably butchering that name.
Something Greek, yeah, no, I don't remember either.
But yeah, so it took them 20 years to make this mouse model, and I think it's the most impressive piece of genetic engineering on a mammal that I've ever seen. So they took megabases of DNA in the B cell locus, and they basically humanized much of their B cell immune systems. Right, and actually, when you look at Regeneron's performance, and he showed this data, if I got this right, then 8 out of 8 drugs that they have built over the past over X many years have been successes. They've gone through clinical trials and have been market successes. And they've all been antibody-based drugs. And that's 100% success rate in an industry in which the average failure rate is 90%, 90.6%. And that's just insane. And, in this presentation, he said, look, the reason why we can do this is because we have this model where we know it replicates the human biology here. And secondly, we have another way to use like human genetic data to say, is this a good disease where the model actually replicates the disease or is it not? And if it doesn't replicate the disease, we don't go after this. So they've done a very good job. It took them a very long time. But I think that's one example here. The other example, and it's a very interesting talk that I saw by the author of the Irooms law. I don't know if you know the law. It
Yep.
basically says that like drug development costs have increased like twofold every X year. Um, and he did a basic mathematical analysis on what's the distinction between screening more molecules versus having a more predictive model. Right? And apparently, what he found was if your model predicts outcomes 1% more often, in terms of what your disease actually does, then it has the same effect as screening 16 times more drugs. Right? And that's just insane. So, yeah, I think actually I really, respect and support what Regeneron did. And maybe they're gonna, run its course at some point where it's gonna run out of targets and whatnot. But yeah, so far they seem to be pretty well.
Yeah, yep. Um, yeah, I mean, like, on the in silico side, I think that um that applies as well. And I think it's very, very interesting. A lot of people would just run very simple docking kind of simulations. And then if it gets a good score, they say, okay, let's move to the next phase or whatever. There's so many more things that you could do, right? You could run a variety of different algorithms. You can run with, different conformations of a protein. You can run molecular dynamics or free energy perturbation or other, kind of, more detailed models or whatnot, right? So, in my mind, like just running through, a billion different compounds and then picking it best, right? Like doesn't make that much sense. There are so many other things that you could do, right?
Mm,
For maybe a little bit additional effort that would give ideally much, much better predictions. And I think the failure rate in silico screenings is much higher that than in biological, tests, right? Assays.
mm,
It's probably like, 95 to 98%. I think
mm,
like, the standard must be, around the 2 to 5% kind of success rate where you find a molecule that seems to work and then you test it experimentally uh, with with an assay and then it seems to kind of give
mm,
you something interesting. And I think part of the problem is precisely what you said, right? Like the predictive power of individual tests is just not good enough.
mm, mm. Interesting. But actually, I would argue that here's where the data science nature of biology is going to come into play, which is in silico right now is probably not as good as doing things in vivo and probably won't be for a while, but eventually it will be as good and then better. Right? And I think the delta between where we are now with in silico and where we could be and where we ultimately want to be, which is 100% prediction and like full simulation of all biology on everything, is high quality, reliable, standardized biological data. Yeah. And fundamentally, we just need this because, when you do the basic background of math of how many different states a cell could be in, or like if you take all the variables together, then you can't run a physics based simulation, at least right now, where you just try to mental engineers. There's just too many chaotic variables. Yeah, engineers like trying to predict weather to a certain extent, but much more difficult. So you need to measure a lot. And then the question is, well, how do you build up the infrastructure that allows you to get this data to eventually, deprecate physical testing altogether? And yeah,
Yeah, yeah. And maybe going back a little bit, right? Like there was another thing that you said that I thought was very interesting, which is that you are building a tool that will enable scientific discovery, faster with higher replicability and with more throughput. And I really like that idea. I think that is, in general, not a lot of care or attention for building tools
mm,
um, in the life sciences. I remember reading this story about like there was the one engineer that they had during the Human Genome Project. It was basically, matching all the kind of all the reads, together or whatever. And then he built some tool. I mean, I don't know, like bioinformaticians would know this better than I do. But I remember reading about that. And it was like, yeah, they had this, multi-billion-dollar project and like all sorts of, doctors and so on and so forth. And they had like this one guy that was kind of like, hacking together, script to match up the
mm,
data.
yeah,
That's just, it's crazy, right? Like, why didn't it have a whole team? Like this was obviously, a very important part of it, right? That would determine, success or failure or not.
mm,
But I guess, that is a little bit different than in the computer science world, where you definitely see a lot of care for people building tools. So
mm,
I sometimes tell people what, like if you look at, say, mechanical engineering versus computer engineering, right? Like a mechanical engineer is, I think, maybe a little bit more productive than they were, 30 years ago, right? But like, still you cannot design a new car, in 3 days, right? It
mm,
doesn't work like that. You still have like yearly car models or whatever. Whereas if you wanted to build uh, a website, with, like basic stuff, logging, storing some data, saving it, whatever, right? Like so many people could just build that in one afternoon,
mm,
right? And of course, the reason is because we've spent decades building tools upon tools upon tools upon tools, right? So, somebody built operating systems and databases and compilers and so on and so forth. So now an average engineer can just go and import those libraries and it's like, getting more work done than 1,000 engineers, 50 years. That has not translated into life sciences, and I mean, I just can't understand, why.
mm. Yeah, I would agree. And I think that's exactly where we need to be, which is better tools. And in some ways, the genomic space has gotten there. And more recently, the transcriptomics space. Proteomics is just making that transition where you have standardized digital tools that are cheap and easy to run and kind of standardized workflows to analyze them. Um, Animal testing is not even close, not hasn't even started, right? And arguably, the other side of it, the engineering side, also is like, really very early. Like writing, we're writing machine code right now in some ways. But I think with regards to genetic engineering, I'm actually somewhat hopeful that we're gonna do the same type of sigmoid curve as we've done with other systems, because to a large extent, genetic engineering right now is limited by existing genetic engineering tools. So if you have a better CRISPR, well, now you have a better tool to build an even better CRISPR, right? And so on. And so that's one aspect, and then there's the delivery aspect as well, which is as the delivery gets better, every single experiment gets easier and cheaper to do, and therefore people will want to do, more of it, and so on. And at its end, final destination, getting new genetic material into your body should be like a totally mundane, everyday thing that you do that you just don't even notice, right? And it's definitely.
A 3D printed, right? You go upload instructions to the 3D printer, half an hour later, you have your little plastic object, right? So you need a DNA printer and lipid nanoparticle kind of, manufacturing machine. You type your, ACTA or whatever, right? And then, you get your lipid nanoparticle with your, genome, on the other side.
Yeah, exactly, exactly. Yeah, and it's just at the end of the day, it's fundamentally it's information processing, right? It's reading biological information, writing it, and you're still dealing with uh, it's, for example, DNA. Sure, it has a physical structure, but it doesn't necessarily need to always have a physical carrier. Like you don't always have to, like, theoretically, you don't always have to, deliver a bunch of molecules just to modify DNA, for example, because it's an information carrier. Just as you don't need to, send a USB stick every time you need to send an email. And I think that's where we are converging. But the tools and the tools to build the tools to need to get there aren't there. And I would argue that that's, as of today, the biggest leverage point that an individual can do to get to that, to get to that place, and therefore to get to the place where we're gonna have these like insanely impactful new systems to engineer longevity and everything else.
Yeah, yeah, I really like that. I mean, that's how you get to exponential growth curves, right? An exponential growth curve comes because, a 1% improvement on one side, causes a 1% or 1.5, 3% improvement somewhere else, and kind of, compounds over time, right? And then, suddenly you have processors that are, a billion times more powerful than whatever we used to, fly to the moon or whatever, right? Yeah. I mean, if you want to speculate, how would that look like in the life sciences? Let's imagine that we have, you know, I guess, magic tools that help us build those therapies, and then we know that they get delivered across all the cells, and then we have a way to test that in animals quickly and cheaply, right? Then what happens next?
So first, it's about doing things better physically, like moving atoms cheaply, more faster at a more higher throughput. And that's what we're doing right now. So that's, the first product that we built is basically just this smart lid that tracks animals, uses computer vision and AI to automatically pull out their health data. So now what you could do is you don't need to have a human measuring it anymore. You can just, have the computer measure like what's the health of the animal, right? And so that removes some of that moving of the atoms. And the same thing is happening on the right side or on the modification side, which is, you know, better delivery tools, better CRISPR tools to modify these atoms in the DNA stuff. Right? So that's one. Now, as these two large aspects of biology, which is the understanding of it and the prediction and the modification of it or the treatment, converge or improve in their evolution, then the next phase of this is modeling. You know, you model more and more systems based on the better data that we have, and less you do these actual, you know, physical experiments. And of course, the ultimate convergence of this is, again, as I said, full fundamental understanding of everything in biology, where to like you run a program that says, look, I want to treat this disease, and here's the drug, and it's going to work 100%, no questions asked. And you just put the human to kind of confirm, but like it's going to work. And that's like the equivalent of, I would say, well, maybe not quite, but a lot like how chip design is today. They don't do physical experiments. They simulate like hell, and then there's something that will work that they make the fab for, and then that's how it's gonna run, right?
Yep.
And so that's the
Many other things in computer science, right? Like, any code, right? Like you write your code and then you compile it and then you test it and then you know that when you deploy it, it's going to work. It's not 100%, but it's definitely better than if you don't have tests or if you don't have compilers, right?
yes, 100%. So that's one area. Um, and then on the other area, we have the ultimate ability to control biology, which like right now, like we can't even do gene therapy. Like we can't put a small piece of DNA into a body for most things. I was like the first one, or really one of the first ones to show that you can even get a small piece of DNA across the body for most tissues. That's like new. So that's how basic we are right now. And then we get to a point where, well, now it's kind of safe and it's at least replicable, where you can do it for most things, but it's maybe still expensive. But now you can kind of deliver across the body. And of course, you've been able to do it in a cell already for a long time, but doing things in a cell alone isn't going to get you to where you need to be because you're a full organism, right? And then iteratively, the cost is going to have to come down, and the size at which you put in these pieces of DNA is going to go up, and the frequency and the like all the barriers, which is basically immune reaction, readability, cost, size, stability, and predictability of how something functions in the cell, like all of these dimensions are going to have to lower, and probably they're going to do this evolutionarily, right? And I have, some even more radical ideas there that I'm not going to go into right now. But when you run this to its course, then you know, taking something like a small molecule generic today isn't a big deal. Like, maybe it's going to cost you like, I don't know, a few dollars per dose or $10 per dose. But
Usually they are cheaper to synthesize. Yeah.
exactly, because people have figured out now for hundreds of years of how to do chemistry and so on, how to purify and whatnot. It's not about the cost of the molecule itself.
Yeah.
Like, it's nothing. Like, these atoms cost nothing. Right? It's about the manufacturing process. And like a good example of that is, for example, the fact that right now, as of today, we are still using animals to make these gene therapies. Like, a cell that is humans, that doesn't matter. It's an animal that is making the gene therapy, that is making the protein. So you have to grow out the cell in a vat, then you have to purify.
Chinese hamster ovary cells, right? I guess
Yeah.
that's like what food.
Is a big one, right? It's like, well, like using animals to plow your field, or move you around. Yeah. Um, and that's gonna go away fundamentally, and we know we can do this. And yeah, I think at the end of the day, modification of like just purely modifying DNA in the body is going to be as simple as uploading code to your computer. Like, that's the fundamental limit. And why am I talking so much about the DNA? Well, it's because of two reasons. First, it's the only thing that you can do stably in the body. And as I mentioned, particularly with longevity, we know that one thing is not gonna work. You have to do multiple different things. You have to keep on building, right? And every time you solve something new, let's say we solve cancer, cardiovascular disease, and dementia right now, maybe people would live to 150, but then something new comes along that isn't the problem right now. So then you have to solve this, then you have to solve that, right? So in some ways, it's a never-ending cycle where you only, you can just push the boundary back further. But in order to do that, you have to have something that is stable, where, look, I made this modification, that is stable, that's my new baseline. And now I make another one. That is stable, that's my new baseline. And now I make another one, and another one, and another one. And that's how evolution got us where we are right now. Literally, that's how it did it, right? So you need DNA for this. There's nothing else that can substitute, including replacement. And the second part is, well, DNA is actually the ultimate drug, right? DNA can make, RNA can make protein. You can make a protein that has, it's 1,000 amino acids. Each location, each amino acid could have different, amino acids, right? So if you put this together, that's 1,000 to the power of, no, 20 to the power of 1,000 on top. Which is like, more than, I don't know, atoms in the galaxy or something like this. Like,
More than photons in the universe. Yes, it's a big, big number. Yeah.
so practically, it's an unlimited space. Now, of course, uh, there are certain pockets which are definitely not going to work. So it's not like it absolutely is unlimited, but practically you can make anything, everything that you see today in the world, including our technologies, out of biology. Right? It's like the ultimate molecular synthesizer. And so why, like, there's no reason for anything else when you're at that stage. Right? So yeah, that's, I think, where it's going to head.
Yeah. I mean, it's that is very exciting, right? I think so far, like all the gene therapies that have been approved are maybe relatively simple compared with what you described. It's like, well, we know that there's this protein that misfolds because there's one error. We just fix this one little thing on one small subset of cells, and that cures the disease, right? Like that's the what is it, the sickle cell anemia, for example, right? Single mutation, you just need the hematopoietic cells, I guess, in the in the femur, right? And then that's it. You fix it there, and then that cures the disease. But of course, like you could design proteins that break down, kind of amyloids that form in the body, for example, and then you could use a gene therapy so that your body suddenly learns how to break down those amyloids, and then amyloidosis is no longer a disease for
Mm,
that person, right? I think that would be pretty fantastic.
mm, exactly. And there's one thing that I would argue, particularly with the experience of COVID that mankind went through, which is as these tools get more powerful, so will the ability to engineer hugely bad human pathogens get easier. And if you run these probabilities to to its natural course over time, then it'll approach 100% likelihood over sufficiently long time that there's going to be someone at some point during history that's going to generate a a pathogen that's going to wipe us out. Right? There's only one defense against this, really, and that defense is being able to generate the antigen, or like the defense against this thing faster than what it can spread.
Yep.
But you can only do that if you're very fast at genetic modification locally, because otherwise, guess what? Someone's going to have to do the study, manufacture it, ship it. That takes too long. We saw this already now. So I would argue that fundamentally for us to survive as a species, survive this particular threat, we have to get there. That's it.
Yeah, um, that's not a happy note to end the podcast. So maybe we'll switch to something else to not leave it there for people to kind of have a nightmare at night.
Maybe
But
we will take action to become genetic engineers.
yeah, yeah, no, for real, right? But I mean, it's kind of a scary thing, right? But yeah, no, I think uh, sometimes people are scared of like climate change or or nuclear war or whatever, right? But I think somebody some rogue lab somewhere doing gain of function of a virus, right? It's definitely a lot more scary. I mean, we definitely saw millions of people died, and so on with COVID. Um, Maybe go back to something happier, right? Like, so
Yeah.
at which stage, are you in the company, right? Like, how long until we have this, fully automated, cage when somebody can just, go ahead, buy it and then, have some animals shipped to the lab and then
Mm.
you just plug it to the electricity and let it run, like
Oh,
how.
we're already there. We've been there now for more than half a year. Yeah.
Nice.
So yeah, the system's working in the hands of close to, I suppose, 200 users
Right.
right now, and we are slowly scaling and now learning our lessons and expanding it. And yeah, uh, it's found a home in many many other places beyond longevity, which I was both glad and surprised to see. But yeah, that's the first leg of automation, where it's automating the data collection. And then once we are there with this, and this is a solved problem, then the next step is automating the physical movement and handling, and just like increasing the throughput at which we can handle these animals. And then it's more robotics. And what's up there?
Congrats. And then the surgery at some point, maybe the extraction of the tissue, or is that too hard for that?
Well, actually, that part is not that difficult to do in terms of, well, it is difficult to do if you want to automate the harvesting of the tissues, because that takes a lot of, it's very actually very computer vision, like finance problem. And it's like probably more difficult to replace these technicians than it is to replace, I don't know, like a CEO, just because of the complexity. But this is not the most time consuming part. It's much more about process engineering. And even in my PhD, I came up with a process that was, at the end, it was like reasonably good. I would be able to do them, even on my own. But that just takes, honestly, just basic automation, and I think that's not that difficult to build, honestly. And there's people already doing this right now. So I think it's the problem that I don't need to solve. And by the time I'm done with this, and if still a problem, then I will solve it. Um, yes. Uh,
Awesome. Yeah, I mean, and robotics is definitely moving very, very fast, right?
mm,
I think what was the latest one? The Unitree, I think it's called Unitree G2. One of these kind of humanoid like robots that walks by itself. It can carry boxes and so on.
mm,
It's not as typical, probably over the next 4 or 5 years, we're going to see robots that can do the laundry and wash the dishes, and then at that point, that's going to be the kickoff of the robotic revolution, right? Because if you can build something that does the laundry and the dishes, you can sell it for $20,000 today, right? Um, Anyway, but yeah, so then going from there to, like, I guess, moving animals from cage to cage, right? This it's not a big jump.
Yeah, and I think again, robotics historically has been successful if it's very narrowly built and you know the problem very well. And I think animal studies is a good way to do that. And you don't need to automate like the very finest things at first. They're not honestly they even the most time consuming parts. But yeah, but the big thing with robotics though is right now an aged animal is very expensive. It's about $600 or $700 per animal. It means that for you to run your 100 animal study, it's $60 to $80,000 right off the bat up front, right? And all of this comes from the fact that almost every step of the care of the animals right now is done by a human hand. Therefore, it's expensive to get animals to be aged to the point where they are already old. And that's why we build robotics. One of the big reasons, because when you automate that basic care aspect, you can also lower the cost, and therefore you can lower the barrier of just doing more of the studies.
Nice. Awesome. Um, cool. Uh, I think this is a fantastic discussion. We touched on a lot of stuff. Time went flight. I
Yeah.
think we actually, our meeting was ended like 10 minutes ago. Sorry if I made you late for the next meeting. But it was a pleasure. So thank you so much for taking the time to
Yeah.
chat today.
Yeah. Like it, Javier. Always, always. Right.
Cool. Well, if the last thing I guess is if somebody has questions or whatever, I guess they can reach out to you, LinkedIn, Twitter, your company website. What works best.
Yeah. Yeah. Email me at michael@oldenlife.com. That's also good. Yeah.
Awesome. Thank you so much, Michael.
Pleasure.
It was a pleasure. Best of luck with whatever comes next for the company. I'll keep an eye, and then love to meet again. Chat in person. Any of the I guess conferences that are coming up.
Mm. Yeah. Likewise. Likewise. And same to you. Best of luck for the next stages of your very exciting company. Yeah.
Thank you so
Okay.
much, Michael.
Thanks. Yeah.
Bye.